Okay, so we're going to make a start now. So just a little introduction. My name's Madiha. I'm a Year 13 student and I'll be your biology tutor for today. So I do AQA at my school, so this whole thing is going to be based off the AQA specification. So you may need to cross-check with your spec to see if you do the similar things. But so we'll be going through DNA, genes and chromosomes. So just some safeguarding, just to make sure that you're all aware. Microphones must remain muted at all times and cameras should be turned off. If you are under 18, please make sure that a parent or guardian knows that you're attending the session. And if you do have any questions at all during the whole meeting, just type them into the chat. I'll get to them either at the end of a section or at the end of the whole topic. This topic is quite short actually, so I'm expecting to finish quite quickly and then we could do some exam questions instead of doing them on the chat, just because this topic is quite short. But yeah, so this is the specification. This is literally all we have to cover. So I've split it into 4 different things. So the first thing is essentially the differences between prokaryotic and eukaryotic DNA. So we'll be going through that. And then we'll also be going through what a gene is and what genes code for, essentially where they are in the DNA. We've also got to go through the genetic code. And we also need to go through differences between introns and exons. So, with that being said, we're going to go straight into it. So we've got these 4 sections. We're going to be going through prokaryotic versus eukaryotic DNA first. So what are the similarities between eukaryotic and prokaryotic DNA? If you can't remember from GCSE or you just want a bit of a recap, essentially eukaryotes have a membrane-bound nucleus, or prokaryotes do not, so that's the difference between them. So in terms of their actual DNA, it's really similar. So in terms of their structure, we have got the DNA nucleotides containing deoxyribose, phosphate group, and a nitrogenous base. So if you remember from the very first topic, you've got the nucleotide structure looking something like this. In this, we've got the phosphate group here. We've got the pentose sugar, so this would be deoxyribose. And then we've got the base here. So the bases can either be adenine, guanine, cytosine, and thymine. So this is essentially the makeup of a nucleotide. And in every single DNA, so in both eukaryotic and prokaryotic DNA, you've got both of these, you've got all of these nucleotides looking the exact same. And in terms of how they actually connect, so we've also got nucleotides joined together by phosphodiester bonds to make a polymer chain. This is the same in both eukaryotic and prokaryotic DNA. So I've just got a little demonstration to show. Essentially, if we've got another nucleotide, you can see the phosphodiester bond form between the two. So we've got pentose sugar and the phosphate group. So a phosphodiester bond forms between these two. So if we add another nucleotide, you can see this thing start to join. So essentially, in both eukaryotic and prokaryotic DNA, you've got this same build-up, you've got the same makeup of how DNA is made. It's double-stranded, it's the exact same thing. So that's essentially the exact same thing in both eukaryotic and prokaryotic DNA. But in the exam, they'll ask you what are the differences between them. So when it comes to differences, with eukaryotic DNA, it tends to be longer, and prokaryotic DNA tends to be shorter. This is just keywords you need to know. Sometimes it's better to say longer and shorter. Sometimes they let you just say long and short. I say just use the to make it a bit more specific. Eukaryotic DNA is linear, prokaryotic is circular. So you might come across the term, plasmids. So sometimes in prokaryotic DNA, you've got DNA kind of forming little plasmids like that. In eukaryotic DNA, you don't have this. It's essentially just all into chromosomes, which we'll talk about later on. And eukaryotic DNA is associated with histones. So these are the type of proteins, which we'll go through in a bit. And prokaryotic DNA is not associated with histones. Another thing to just note is that sometimes the exam will ask you things about, describe the DNA in chloroplasts or describe DNA in mitochondria. So these types of structures are structures that you would see in eukaryotic DNA, right? However, when it comes to actually describing this DNA, you would compare them to prokaryotic DNA. So essentially, the DNA in the mitochondria and DNA in chloroplasts would be shorter, they would be circular, and they would not be associated with histones. So just key thing to remember when you're asked to describe mitochondrial chloroplast DNA, you would basically use the exact same description for prokaryotic DNA. So it's a shorter, circular, not associated with histones. But to actually understand this, so this is something I used to struggle with at the start of the year, like understanding the makeup of DNA. So essentially what makes up a chromosome. So like we said in eukaryotic DNA, it's associated with histones. What does this actually look like? So here we've got a picture, I've got it from Cognito. So here we can see, why has it done that? Okay, here we can see the DNA bases, right? These little things. So this is a whole DNA molecule. All these bases joined together, making that double strand. It says here DNA molecule. This is wound really tightly and it's gone really thin. And it starts to wrap around these kind of circle-looking structures. These are called histones. So these proteins are essentially what the DNA wraps around. So it's wrapping like tightly around all of this. And as it gets even tighter and tighter, it forms this DNA histone complex. You don't need to know about this. It's just to help you like understand it. It gets wound tightly even more, so it's like really thin. This is what forms chromatin, and this is essentially what you see in like mitosis and things. And this winds down even tighter and it fits inside like this, inside the chromosomes. And this is how DNA essentially looks inside eukaryotic cells. Because they're wrapped around these histones, you get these kind of chromosome shapes. So if you go back to prokaryotic DNA, essentially they're not wrapped around these histones, so they're just going to like be left around like this and just form like circles, plasmids and things like that. So hopefully that makes some sense. Again, I've got just another diagram to just show again in case you didn't understand it. So we've got the bases here, all these DNA bases. This forms the DNA helix, so this double-stranded thing. Again, it's getting wrapped tightly, really small around these histones. This is what forms the chromatin, and eventually it forms the chromosome. So, by the way, if I'm going too fast, please let me know in the chat, just because, I don't want to like rush through this topic. But yeah, so it forms the chromosomes. So if you're asked in the exam what makes up a chromosome, you would say something like, it is a double-stranded DNA molecule, so this here, and it's wrapped around its associate protein, so the histones. That is what makes up a chromosome. That is what a chromosome is. It is the DNA helix, the DNA molecule, wrapped around all of the associated histones. And so that essentially covers prokaryotic and eukaryotic DNA. And now we'll be moving on to genes, which again might be a little bit more complicated just because I used to always get confused with what a gene actually is compared to DNA. So we've got another diagram just to show that. So here we've got a cell, right? So we've talked about the chromosomes having all this like little DNA molecules inside. So look, we've taken the DNA out. The chromosomes are all in their cells. If we look inside that and we see that DNA, this, just a small section of the DNA here is essentially a gene. So a gene is a base sequence of DNA. And I've seen it in mark schemes, they specifically want you to say base sequence because, it's the basis. So you've got like A, G, T, C, it's like a whole sequence. You could have a sequence like this. You could have a sequence like a really long sequence. Whatever the sequence is, that is the gene, okay? And genes can code for the amino acid sequence of a polypeptide or a functional RNA. So again, it's important that you say amino acid sequence. It's not going to just code for one amino acid. It's not just going to code for a polypeptide. It is the amino acid sequence that makes up that polypeptide. And if you're a bit unsure of this, make sure that you go back to topic 1 because it talks about amino acids and polypeptides. So yeah, just make sure you know that. And then when it comes to a functional RNA, this is essentially molecules other than mRNA that perform special tasks during protein synthesis. So you've got things like tRNA, which is transfer RNA, and rRNA, which is ribosomal RNA. These come up in later topics, so you don't need to know them all for this, but it's just essentially you just need to know that a gene is a base sequence of DNA that codes for these things. And again, another key thing to just remember, it codes for. Genes don't actually produce these things, they don't just make them, they just code for them. And so yeah, that's what a gene is. So what, wait, also I forgot to say, it's not in this topic, but I don't see it anywhere in the specification, like when you're meant to learn it. So I've put it here, just in case. But if you don't remember from GCSE, just make sure that you know that an allele is a different version of the same gene. So you can have a gene here, so this gene here, let's say this little gene thing here, this could code for, I don't know, hair colour. This would code for like brown hair. You could have a different gene, so in a different chromosome, again in the exact same place, that gene could code for like blonde hair colour or something. So they would be alleles because they're different versions of the exact same gene. They're coding for the same amino acid sequence of a polypeptide, the same polypeptide, which is like hair colour, but they're different versions, they're coding for two different things. So that's just something to be aware of. But now we're going to talk about where they're located in the body, in the chromosome. So, key term to remember, locus, or plural loci. A locus is essentially a particular fixed position on a chromosome that a gene occupies. So, I've just drawn a little pair of chromosomes here. Looks a bit weird, but so this is one chromosome, this is another chromosome. And here, I've drawn this kind of red thing. So, this is essentially a gene. There's a gene on both chromosomes and they're in the exact same place, okay? So this is telling us that these are homozygous alleles for the same gene. Homozygous mean, they're the same thing. So the same alleles for the same gene. How do we know this? Because the genes occupying the same loci, they're in the same position on the chromosome. So this gene, is over here. This gene is they're in line. So you know it's the same gene. And because they're the same colour, then you know that they're essentially just the same allele. Same allele. So if I give a different example, this one, essentially you would know because they're at different places, okay? They're not occupying the same loci. Therefore, they would be two different genes. Why? Because, again, they're occupying different loci. One's up here, one's down here. Two different colours, they're two completely different genes. Like over here, again, if you go back to hair colour, these could both be coding for brown hair, let's say. Over here, this gene could be coding for, black hair. This one would be coding for blue eyes. Okay, they're two completely different genes. And then we've got, again, two genes occupying the same loci. So you know it's the same gene. However, they're going to be different alleles because they're two different colours. So they're heterozygous alleles. These terms, heterozygous and homozygous, do come up later on. And they're not in this topic, but I just kind of included the example now just to, just open your eyes to it, just so you're aware of what they are. So yeah, the genes are occupying the same loci in the exact same place, but they're two different alleles because two different colours. So maybe they're both for eye colour, one's for blue eyes, one's for brown eyes or something. So there's just some examples of how genes are located on a chromosome. So hopefully that makes some sense. But yeah, that was a really short section of the topic. And now we're going to move on to genetic code. So this is where it gets it's a bit more content heavy, so make sure that you're understanding this and you're writing this down. If I'm going too fast, please tell me in the chat. So, what is the genetic code? So the genetic code is a sequence of bases, essentially a triplet, that code for an amino acid. So I've got an image here. You can see this is the whole gene, let's say. And this specific triplet, so they come in pairs of three, they come in triplets, yeah. And each one codes for one specific amino acid. So you remember how we said that a gene codes for the sequence of amino acids? One triplet codes for one amino acid. That's just amino acid because I can't bother to write it all. So sequence of amino acids is a gene, so we can say this whole thing is a gene, but a triplet is one amino acid. It codes for one amino acid. Again, here it's codes for. It does not produce it, it just codes for it, and it's produced later on. So there are some features that we should be aware of the genetic code. We can describe the genetic code as being degenerate, universal, and non-overlapping, which I'm going to go through now. So the genetic code is degenerate. What does that mean? Essentially, each amino acid is coded for by more than one triplet of bases. So what that means is, let me break it down. So here's a table of all of the amino acids that can possibly be created. So, we've got all of these things, these are different amino acids, right? And then, how do I rub this out? Okay, never mind. So in life, there are 20 amino acids. So in this table, there are 20 because these are the only 20 amino acids that exist. We've also got 4 DNA bases, right? So, and we've also found out that it takes 3 bases needed for 1 amino acid. So if I just bring some maths into the equation, I don't actually take A-level maths, but you know what, we'll just go for it. So let's take it like this. So let's say that there was only, so we know there are 4 different bases. Let's say that only 1 base was needed to create 1 amino acid. That means that we'd only have 4 amino acids coded for. And obviously this isn't enough because we know there are 20 amino acids. So just 4 bases by itself wouldn't be enough to code for all of the amino acids. Then we've got, let's say it takes 2 bases to make 1 amino acid. If we had 2 bases for an amino acid, only 16 would be coded for. This still isn't enough because we know there are 20 of them. So then let's say we have a triplet of bases coding for an amino acid. And we can see that there are 64 that are coded for. So 64 is quite a lot considering we only need 20. So essentially what this means is there are 64 possible combinations that the triplets can make. And so with that, 20 amino acids will be made, and they can be made like multiple times. So as you can see in this table, let's see this here. ACU, for example, it makes this THR. ACC also makes THR. ACA also makes THR, and ACG makes THR. You kind of see what I mean. So multiple triplets can make an amino acid. So that's essentially what degenerate means. It means that more than one triplet of bases can make an amino acid. So each amino acid is coded for by more than one triplet of bases. And that's what degenerate means. So moving on to universal. Universal, the genetic code being universal essentially means that the same triplet of bases codes for the same amino acid in all organisms. So this is more straightforward. So again, we've got the same table. These 20 amino acids are essentially coded for by every organism. They always make these same amino acids. So for example, GCG in a dog. So if we just look at GCG, we've got where's G at? G. So as you can see, GCG would make this, right? ALA. So GCG in a dog would code for ALA. How about in a pigeon? In a pigeon, it would also code for ALA. It's the exact same thing. In a fish, it would exactly code for ALA. And a spider as well. Like, it will always, in every single organism, if you've got the same triplet of bases, it will always code for the same amino acid. So yeah, essentially this is something to remember, and we'll go through the advantages of this in a bit, because each feature of the genetic code is really useful for different things. So the fact that every organism will code for the same amino acid is actually really useful for, things like genetic engineering, but we'll go through that in a bit. And the last feature of the genetic code is that it is non-overlapping. So what does this mean? Each base is part of only one triplet, so each triplet is read as a discrete unit. So here we've got this whole gene. As you can see, it's got a lot of long letters. Essentially, each triplet is read by itself. It's read discretely. So these 3 are always read, then it's these 3, then it's these 3, then these 3. It's not going to be like 2 and then like 4, and then they're always read by itself. So this is good because each triplet is seen as its own thing. So when it comes to things like mutations or different things, it will be okay, because if only one thing is read, it means that other things won't be affected. So before I go too much into it, we'll talk about it now actually. So what are the advantages of the genetic code? So the fact that it's degenerate. Degenerate meaning that more than one triplet of bases can code for one amino acid. So if a gene mutation occurs, the triplet of bases will change. the triplet may still code for the same amino acid and therefore have no effect. So essentially an example would be, you have ACC, and if that C changes to a G, obviously that's a mutation. However, if we look back on our table, let me just go through, over here. So in our table, what was it? ACC and ACG. So we've got ACC and ACG. Both of them are still coding for the exact same amino acid, which means that obviously it's fine that the code is degenerate, because it means that there'll be no effect on the organism, because even if there's a mutation, the same amino acid is still coded for, so it would have no effect. So that is a good advantage of the genetic code. So an advantage of it being universal is that, genetic engineering and modification is possible. So you see examples later on in the course about inserting the human gene for insulin into bacteria. Because they make the same amino acids, it's easier for it to reproduce, it's easier for it to actually be able to produce the same things, because it's the exact same triplet of bases creating the exact same amino acid. And an advantage of it being non-overlapping is that if a point mutation occurs, so basically the change in one single DNA base, so like here, if we've just got one changing C to G, only one codon will be affected, and so only one amino acid will be affected. So here we've got a little gene here, we've got 3 triplets, and as you can see, one thing is being changed. So if only the first codon is affected, only one amino acid is affected, because each codon, each triplet of bases, is only read by itself. So if only one changes, the rest is still read by itself and it's still fine. So that's a really important thing about the genetic code being non-overlapping, because it means that if one mutation occurs, the whole sequence isn't affected, because only one triplet of bases would be affected. So hopefully that makes some sense. And now we're going to be going on to the last part of the spec, which is introns and exons. So what are introns and exons? So we've talked about genes, we've talked about what they look like inside DNA. So introns are sections of DNA that do not code for sequences of amino acids, and hence polypeptides. Exons are sections of the DNA which do code for sequences of amino acids and polypeptides. So if I just bring up this example here. Here we've got a gene. We've said that a gene can code for a sequence of amino acids, and we've said that a gene can also code for a functional RNA. So here we've got this whole thing, it's coding for something. However, if we split up that gene, we can see we've got exons and we've got introns. The exons are the things that actually code for these amino acids. So this exon would code for this small sequence of amino acids. Then we've got intron. This whole intron will just not code for anything. It's just a non-coding region. Then we've got an exon. This is what's coding for something. This is the coding region, okay? And then we've got another gene here. And another thing it says in the spec is that in between genes, you've got this little, I've added this little purple thing in. This is sometimes known as I'll just write it here. It's called a non-coding multiple repeat, or sometimes a region, but I think it's better to just know non-coding multiple repeat. These are things in between genes that do not code for things. So essentially, don't get them confused with introns, because again, they're both non-coding regions. However, introns are within the genes, introns being in, and non-coding multiple repeats are in between genes. So between 2 genes, you've got these non-coding multiple repeats, and they do not code for a sequence of amino acids or a sequence of polypeptides. They do not code for anything. So that's essentially the structure of genes in a whole genome or whatever. So yeah, and another thing to just remember is introns are only found in eukaryotic DNA. They're not found in prokaryotic DNA, because remember how we said that the eukaryotic DNA is longer, so obviously they would have more space, so they can have these introns fit in. But yeah, so if you go back to that slide from near the beginning, I've just added an extra one here now. So eukaryotic DNA contains introns, and prokaryotic DNA do not contain introns. And again, it's the same for mitochondria and chloroplasts. they do not contain introns because it's the exact same as prokaryotic DNA. So yeah, I think that brings us to the end of actually all the content. So does anyone have any questions on the content? I know we've got half an hour left, so I've got some exam questions that we can do. So does anyone have any questions right now? If you want to type them in the chat, if you want to raise your hand so I know that you're typing in the chat, just so I don't move on too quickly. So no questions? Okay, that's fine. We'll go straight on to exam questions. So I've got 4 exam questions for you to do. I think we should spend about like 5 minutes on each one. So I've put this on the board. It's 3 marks. So we'll spend about, we can do like 2 minutes. We can do this in 2 minutes. So if you want to like get a piece of paper or get something, and hopefully you can see that question. Complete table 1 to show 3 differences between DNA in the nucleus of a plant cell and DNA in a prokaryotic cell. So I'm just going to highlight some keywords here and just fill in the table. So at about 32, I'll come back and we can go through the answer. Yeah, I've realised this is a very short question, so is anyone still working on it, or would you like me to just go through the answers now? Still working on it, just let me know in the chat. Okay, I take it that everyone's done the questions, so let's go through it. So first I'll go through my answer and then we'll go through the actual mark scheme just to see any key points that are important, because with biology the mark schemes are quite harsh, so I want to make sure that you guys understand the mark scheme so that you know not to make any mistakes in the future. So we've essentially talked about this at the very beginning. We have got DNA in the nucleus of plant cell being longer. So plant cell essentially means eukaryotic cell, so this is something that we're meant to be aware of. A plant cell is a eukaryotic cell. And it's just giving it to us in the question that the other one is prokaryotic. So you know this is going to be a simple comparison question. So we've got it's longer, shorter, linear, circular, associated with histones and not associated with histones, and then other things you can put introns, no introns. And also I didn't fully specifically mention this just because I don't really like this point, because sometimes they give it to you, sometimes they don't. But you can say that a plant cell would not have plasmids, but the DNA in a prokaryotic cell can have plasmids. So that's essentially the mark scheme. Let's talk about specifically. So yeah, we've got all of these points down. Again, here it says long short You didn't need the but sometimes they really need the so you know, it just depends. And again, the reason why I didn't really want you guys to put this one is because it says do not credit if it suggests that prokaryotic DNA only exists as plasmids. Let's say over here you put no plasmids and put only has plasmids. It would be wrong, because some prokaryotic cells have DNA stored in the what was it called? I think it's called like ribosomal DNA. So it's not just plasmids, it's in other forms as well. So you know, it's better to avoid it if you're worried about getting the wording right. But if you put it, that's fine. Yeah, and another thing to just remember, they're really strict about it, but alternatives must be written directly opposite one another. Meaning you can't write longer, linear, associated with histones, and then write circular, not associated with histones, shorter. They wouldn't have given you the marks. You have to literally compare them right next to each other, which is a bit stupid because you're filling in the table anyways, but just make sure that, you know, if you're doing things like this, you're putting them opposite one another. And then it's just little things here, so do not award if only half of the mark points written. So if you only wrote longer and you didn't write anything here, you wouldn't get the mark. You need both to get 1 mark. It's awarded like this. And yeah, if you talk about it being single-stranded, max 2, because it shows that you're not aware of the DNA, because remember we talked about the similarities, that the DNA is, double-stranded because it's got the same nucleotides, it's forming that backbone together, so we needed to put that. You're not meant to put that. And not being helical, again, we talked about this at the beginning, because knowing the similarities helps you understand the differences, if that makes sense. Like, they wouldn't ask you what are the similarities, but because you guys know from the beginning, we talked about how DNA is similar in this way, how it forms the same DNA, then you know not to talk about it in a differences question, because there's no doubt that if you know what the similarities are. So hopefully that makes some sense. But yeah, any questions about this question, or should we move on to the next question? Okay, no questions. I'm just going to move on. So we've got 2 parts to this question, both 1 marks. Again, I'll give it just like a minute. So we're going to talk about is why is the genetic code described as being universal? And then a quick question, the genetic code uses 4 different DNA bases. So what is the maximum number of different DNA triplets that can be made using these 4 bases? So I'll give it a minute and then we'll go through the answer. Hopefully you've had time to do that. If anyone's still working on it, please let me know. If not, then I'll go through the answers now. So we talked about this in PowerPoint. I've literally used the exact same definition I put up. But the genetic code described as being universal essentially means that the same triplet of bases codes for the same amino acid in all organisms. And this is what gets you the mark. And then here it's just 64. You can just literally just memorize this. If you can't memorize this, or you'd never be able to, just remember that the number of bases is essentially the big number that you put here, so it's 4. And the max number in a triplet is 3, so you've got 3 DNA bases in a triplet, like A, G, C, that's 3. So it's 4 to the power of 3, which is going to be, if you get your calculator out, that's 64. And so that is how you would do that question, if you can remember it. So yeah, if we just go through the mark scheme, some things to note. So yeah, we've literally just put this exact definition. Something key, again, because the mark schemes are so specific, they'll only accept plurals if both the triplets, if you put plurals everywhere. So essentially, if you go back to it, if you say the same triplets of bases codes for the same amino acid, that's wrong. If you put the same triplet of bases codes for the same amino acids, it's wrong. They only want you to put plurals if both of them are plurals. So the same triplets of bases would code for the same amino acids in all organisms. So I think it's just better to just stick to single, just so you're being really specific. If not, then yeah, so make sure that you're doing this. And again, remember when I was saying it codes for? I was being so adamant about this because they're rejecting if you write producing, because technically it's not right. They don't produce anything, they just code for. So yeah, essentially that is that. And then the other question again is just 64. If you remember it, that's great. If you don't, I've explained how to work out, so hopefully that makes some sense. Okay, next question. Does anyone have any questions about that, or can I move on? Okay, I'm assuming no questions. Okay. So we've got a slightly harder question now. We'll highlight the important key information. So scientists investigated the genetic diversity between several species of sweet potato. They studied non-coding multiple repeats of base sequences. So the question is asking, define what these are, non-coding base sequences and describe where they are in a genome. So, and you can see it's 2 marks. So if you already split the question up into the define and describe where, you know how you're going to get those 2 marks. So I'm going to give you 2 minutes to do that. Let me know if you're still working when I come back. Okay, so I think that's been enough time. If you're still working on it, just let me know. If not, then we'll just go into it. Got nothing in the chat, so I'm just going to go through the question. So here's my answer first. Again, you know how we've split the question into 2. I specifically highlighted on my answer to show, obviously you're not meant to do this in the real exam, but just to show you what each thing corresponds to. So non-coding base sequences. So we talked about how you've got genes, and we've talked about how in between these genes, so in your genes you've got introns and exons, and in between them you've got that non-coding multiple repeats. So what do? Well, we said that they are sections of DNA that do not code for sequences of amino acids, and hence polypeptides. And they are between genes. So these 2 marking points that you had to get. So hopefully you got those. And let's just break down the mark scheme. So this is what the mark scheme says. So it's giving you 3 different options actually. So it says DNA that does not code for polypeptides, DNA that does not code for sequences of amino acids, or DNA that does not code for tRNA or RNA. Because if you remember what a gene is a DNA-based sequence that codes for sequences of amino acids or functional RNA. So if you remember that, then we know that because these non-coding multiple repeats are in between genes, we know that they're not going to do what a gene does. So all of these things. So in my answer I picked this one. If you put any of these, you're still right. However, you need to be careful because it's saying that you should not credit DNA that does not code for an amino acid. You can't write an amino acid. Again here, they're really specific about plurals and singulars. You want to say sequences of amino acids or just amino acids. Because if you're saying it's coding for one amino acid, you're assuming that this non-coding multiple repeat is just 3 bases. However, it could be like 56 bases, it could be like 100 bases, you don't know. So you always say sequences of amino acids. So it's just a specific thing. And again here it's saying ignore references to introns. Why? Because introns are within a gene. And this is talking about things between genes. So as you know here, between genes. So yeah, don't say in introns or don't say between exons. These are all within a gene. And we want to know what's in between the genes. So you could also say something like this. I would never have gotten this, but you know, if you've gotten this, then well done. But yeah, it's a really specific mark scheme. So again, make sure that you know your plurals and your singulars. Okay, now we're going to go to any questions about that before I move on? No? Okay, then we'll go on to the last question. Slightly more difficult one, but this is kind of as difficult as it gets if it's just this topic. Again, you can come up in other topics when it's more linked to other things, but if we're just talking about just this specific topic, I feel like this is probably the hardest it would get. But not all mutations in the nucleotide sequence of a gene cause a change in the structure of a polypeptide. Give 2 reasons why. So again, let's just highlight. So it's saying not all mutations cause a change in the structure of a polypeptide. It's saying give 2 reasons why. So make sure that you're reading it. It says of a nucleotide sequence of a gene, so you know it's going to be inside a gene. So what do you remember? Just things to think about. What do we know is inside a gene? What do we know a gene does? Things like that. And then you can start getting your answer. And I'll come back in a minute or two to go through the final question. Okay, so hopefully that's been enough time. If anyone's still working on it, just let me know. If not, then I'm just going to go through the last question. So here's my answer again first. So I put 2 reasons down. I think we'll go through the first one here. So I've put the genetic code is degenerate. So it essentially means that the new triplet may still code for the same amino acid. So a mutation in the nucleotide sequence, it could change, one specific base. It could make ACC to ACG, and this new triplet could still code for the same amino acid. So because it codes for the same amino acid, it does not cause a change in the structure of a polypeptide because it's the same amino acid being coded for. So that's one reason why, the mutation will not cause a change. You could literally just write this, and this by itself, I'm pretty sure would get you the mark. We'll go through that. But I always like to just explain it just in case. And then the second reason was because I told you to think what is inside a gene. The mutation may occur in an intron, and as we know, these are non-coding sections of DNA. So essentially, if a mutation occurs in an intron, it's not going to code for anything. It's not going to cause a change in the polypeptide because it's not coding for anything. It's just a change in something that's not being coded for. So these are the 2 reasons that you put. And if we look into the mark scheme, just to see, it's really short. So yeah, the first thing I put was triplets code for the same amino acid. This is all you needed to put. Or you could literally just put triplets are degenerate. The DNA code is degenerate because the new triplet could code for the exact same amino acid. And the other thing is that it occurs in introns. So you're going to reject codons because the question states within genes these codons are that come later on. It comes in different topics, but a codon is essentially what comes later on after transcription and translation. Inside DNA bases, we call them triplets. So it occurs in introns. Ignore junk DNA. Junk DNA is essentially another word for multiple repeats, but it's not as commonly used. But it says to completely reject multiple repeats, non-coding multiple repeats. Reason being, it's because the question specifically states within genes. It's not saying between, it's saying within. And so we know within a gene, there are only introns and exons. And in this case, we talk about introns because they are non-coding. So if you talk about multiple repeats, then you're wrong. But yeah, that's essentially it. So yeah, I think that brings us to the end of the actual PowerPoint. If anyone has any questions, let me know, type them in the chat. If not, then you're all free to go. Thank you for attending. If you could fill in the feedback form, we'd be very grateful. But yeah, thank you so much.